The group of fungi we work with are called the Ophiostomatales, which may not mean much to most people, but the best example is Dutch elm disease. And many of them are not necessarily disease-causing, but they have the habit of staining wood and lumber, and that causes economic problems in terms of trying to sell the material. I've been collecting them for over 20 years, and we work on their physiology, taxonomy. So why is it interesting? Well, first of all, it's genetically interesting. It's a model system of how mobile elements can evolve or change and behave. They allow you to potentially use them as markers to distinguish between different species or different strains. So having DNA markers allows one to distinguish them more accurately. Another application is that actually a lot of these little mobile elements encode proteins, and any protein that cuts DNA can be used in genetic engineering to generate DNA fragments. And so we are sequencing a lot of mitochondrial genomes for many members of this group of fungi to make a database. So if anything gets introduced, we can recognize it and differentiate it from what we have already here locally. One of my students refers to it as sort of the Russian doll analogy. We have found in some fungi in the mitochondrial genome where there's a gene which has acquired one of these mobile elements. But then that mobile element was invaded by another mobile element. And that one also got invaded by another mobile element. So now we are building sort of a complex mobile element that has really three modules. So basically it's present in the genome. That gene is going to be transcribed to make a messenger RNA, which eventually will be turned into protein. Well, in order to get a messenger RNA that's functional, these inserted elements have to be popped out. And so what happens is the internal member splices first, then the second member splices out, and then the original member splices last. So it's almost sort of this Russian doll analogy, but you're starting from the inside, and you're working your way towards the outside. So one of the projects I'm involved is we're using mushrooms to make sort of styrofoam-like material. We're letting the mushroom, the mycelium, grow on waste, shredded cardboard, agricultural waste. The mycelium is like a pipeline system, but it acts like a binding material. So it breaks it partially down, binds it, glues it all together, but you end up with something that some people like to call biofoam, a substitute for styrofoam, which of course environmentally is costly to make and doesn't degrade. Now for us, a bit more of a humble goal is to develop prototypes for packaging materials. Pieces that protect corners, pieces that can be used to embed like a cell phone. But the whole idea at this point is just to develop different types of packaging material, and hopefully we can get some people interested in adopting them and fund us. As you're working with different people, you never know what you get. But partially, of course, it's to find out how we can make everybody work and make sure everybody's happy in a very simple language. I would say communication is probably the biggest component. Many students come from all over the world. And so they're dealing with probably culture shock. So talking to students, talking to anybody in the lab, find out how they're doing, what's going on in their lives, not to be intrusive, but just to care. I think it's important to be a mentor, that you care for your students. So in terms of science, same thing. I definitely will tell them what my ideas are, but I'm very open to obviously hear what they have to say and what they want to do. So I think it's very open to keep an open line of communication. My role is to be a mentor, not a dictator. You know, the old stereotype of the evil prof. And I think overall we've been lucky in my lab. As far as I know, people seem to get along. I think it's communication and respect that is, I think, the key thing for a lab to work. Because ultimately, if people are happy, they will work better, from a selfish point of view. So it goes both ways. You know, you want to have a lab where everybody feels welcome. You want to have a lab where people are looking forward to come to the lab. Oh boy, younger self. Well, I never give up, always have passion. I think that's probably the two things. I started out farming. That's my background. Me and my dad were farming south of Winnipeg here. And one day I decided this may not have much of a future for some crazy reason. I always found these organisms interesting. And so I kind of stuck with them. So I think it's one of those things. If you like something, if you have passion for something, pursue it. And so that's been sort of my story. There was down times in science. You don't always get what you want, but always keep your eye out to sort of get back to what your passion is. And that's really the only recommendation I can say to people is find out what you like and do more of it and have passion for it.